Okay, I'm Kelly Peña. You are welcome for this conference. I'm from Ecuadendro Tropical and Educational Platform focused on dendrochronological issues. On this occasion, we have our conference about tree research in Bangladesh, where trees may respond to climatic or other environmental stress. Our speaker is the Dr. Mahmouda Islam. She is associate professor from Department of Forestry and Environmental Science from Jawaharlal University of Science and Technology in Bangladesh. Dr. Mahmouda, you are welcome, and we are very excited for this opportunity where you could share about your knowledge related on dendrochronological research. Thank you so much, and you are welcome again. And you can start with this presentation.
Thank you so much. Thank you, Kelly. Now I am sharing my screen first. I need to share my screen.
Okay.
Then I can't because I am disabled.
Yes.
Okay. You
can share your presentation.
Okay. Now I am sharing my presentation. Are you see my presentation? Do you see my slide?
Yes, perfect.
Okay. Okay.
Yes, it's possible to see your slide.
Okay. Hello, everybody. I am Mahmouda Islam. Thank you, Kelly, and thanks for joining my talk. I'm Associate Professor, Department of Forestry and Environmental Science, and currently I am in Bangladesh. Currently, I am working a postdoc researcher at WSU, Washington State University. Today, I will talk about the advances in treating research in Bangladesh, a South Asian country. Before diving the topic, I need to, what is treating research? First, we need to focus what is treating research. And treating research, mainly that the research field that deals with treatings are generally termed as dendrochronology. The wood formed within a year is called annual ring. And dendros means the tree, and logy, then study of, and chronos means the chronology, the time. And dendrochronology is the science of treating. And dendrochronology is already developed or established research field in temperate boreal and Mediterranean forest. But however, treating study in tropical region has not been developed that much. In tropic, there is although a number of studied developed recent number of studies have done recently, but still the South Asian part has a limited study in treating research. According to the Climate Risk Index, South Asia region are most vulnerable in terms of climate change. Bangladesh is one of the 10th most vulnerable country. The dark red color represent this is the most vulnerable country among the 10 countries climate risk and climate risk. That's why we selected. It is therefore very relevant to study climate change and relate that also focus the dendrochronology and treating research. Forest, Bangladesh, there are several forest types in Bangladesh, like a tropical evergreen forest, tropical mixed semi-evergreen deciduous forest, and also freshwater swamp forest, village forest. For the two treating study are relatively very new in Bangladesh. first we made a preliminary screening of 27 species, abundant tree species, is to identify the species forming growth ring boundary. Then we made a thin sections of 27 canopy tree species, which are also abundant in Bangladesh. Out of 27 tree species, the 14 species from fairly distinct growth ring boundaries and 13 species from distinct boundaries, but not a fairly distinct. And they made a different cluster based on their anatomic characteristics, also based on their ring with growth ring characteristics. Then among them, we developed first 3 ring with chronology. This is the 3 species, Cicratia tabularis, Tuna ciliata, and Lagerostomia speciosa. And these are the distribution of these 3 species, and it's mostly in South Asia. These species exist in South Asia. And these are the 3 ring with chronologies. Chronology length nearly more than 80 years. And these are the also some events they showed a similar pattern of ring with index. And this one, they showed a climate sensitivity. Like these 3 chronologies contain a strong climatic signals. Then we can see there, this one, like a temperature mean. Temperature mean has a temperature mean. Cicratia tabularis, Tuna ciliata, and Lagerostomia speciosa, 3 of them negatively impact on growth. That means if the climate is changing, that means it's also effect on tree growth. Also the temperature mean, temperature maximum also have a negative impact on tree growth. That's why our main objective, next objectives was to test whether the climate sensitivity is more stronger in recent period, provided that climate is changing more faster in recent decades. Then we fixed one objectives, like one hypothesis, like did the climate sensitivity of radial growth of selected tree species change over time in response to climate change? And we developed made a regional chronology from 2 sites, a regional chronology of 1 species, Cicratia tabularis, and it's like 2 period. We divide 2 period, like a recent period, and another one is a 1950 to 1985, another one 1986 to 2015. And this 2 period, we can see there is a shift in climate, pre-monsoon, monsoon, and post-monsoon, also in dry season. And this impact on our growth of this species, like a recent time, Cicratia tabularis growth showed a higher negative relations with mean, minimum, maximum temperatures and precipitation during recent decade than the previous, like a 1950 to 85. Then the negative sensitivity vapor pressure deficit in April show the sensitivity, that means negative, it's also increased the sensitivity. And then our another, we can see how would radial growth of selected tree species respond to future climate change scenarios. We can also develop a 3 regression model with Cicratia tabularis, Tuna ciliata, and Lagerostomia speciosa. And then we did the future prediction, different climate change scenarios considered, like a representative concentration pathway, RCP 4.5, 6, and 8.5. Radial growth projected to decline by nearly 20% at the end of 2100 under high emission scenarios. And look at this one, they all decreases, the growth is decreases based on their growth decline during the 2100 growth, all that growth decline. And we also did in Bangladesh, after making the chronologies, we also developed some xylem anatomical measurements, especially the wood anatomical measurement, basal measurement. And first of all, we selected 1 species like Cicratia tabularis diffuse porous species, and we developed ring width chronology, number of vessel chronology, vessel density, also mean vessel area chronology. And then we also tested the climate sensitivity with temperature mean, minimum, maximum precipitation. Dominant influence of temperature mean and minimum, and negative effect on ring width and also number of vessel and mean vessel area. These are the negative impact. Higher temperature increase the vessel density. That means it's also the also we tested the ring porous species, two ring porous species, like one tuna ciliata, another is a Lagerstomia speciosa. But tuna ciliata is a semi-ring porous species, and Lagerstomia speciosa is a ring porous species. But they have a functionally different, like a shade tolerant. Tuna ciliata is a shade tolerant pioneer, and Lagerstomia speciosa shade tolerant. And also the density, wood density, tuna ciliata has a low wood density, but Lagerstomia speciosa has a high wood density. And also the fast growing and slow growing. And then we also developed ring porous species chronologies, like ring porous species means larger early wood vessels and smaller late wood vessels. And then they also showed the climate sensitivity especially for Bangladesh. Interspecies difference reflect in climate response. We found that especially February, March, April, like a spring season, when the growing season starts, the tuna ciliata temperature has a negative influence. Also Lagerstomia, they have a negative impact. But precipitation has a little contribution in the growth, because in Bangladesh we have enough moisture in the soil. And then hydraulic conductivity. Hydraulic conductivity, we also measured hydraulic conductivity from wood anatomy, and three hydraulic conductivity chronologies, like one for Cicrotia tabularis, tuna ciliata, and Lagerstomia speciosa. Then the strong effect on BPD in Cicrotia tabularis and tuna ciliata are affected by both temperature and precipitation. Pre-monsoon temperature had a positive effect of Lagerstomia speciosa. But BPD also has an influence of positive influence, most of the time, positive influence on Lagerstomia Cicrotia tabularis. And however, mean vessel area play an important role in hydraulic conductivity. Hydraulic conductivity is an indicator of hydraulic efficiency. Hydraulic efficiency and this higher mean vessel area lead to the higher conductivity, means higher their growth. Then this also the general trade-off, vessel density and diameter trade-off. It's also we also tested this trade-off, like a vessel density increased with hydraulically weighted diameter decrease, leading to decrease the vulnerability to hydraulic failure due to cavitation. And then after the ring width measurement, then the xylem anatomical measurement, we also did the isotope measurement, like oxygen isotope, also the carbon isotope. We also, like these are also the proxies from we collected from the ring width. Then we developed the oxygen chronology, oxygen isotope chronology, Cicrotia tabularis, tuna ciliata, and Lagerstomia speciosa. And carbon isotopes, for the carbon isotope, we using the Farquhar model to measure the IWE, intrinsic water use efficiency. And decreasing carbon isotope discrimination, increasing the water use efficiency. Also the increasing water use efficiency increased up the 46% over the past 3 decades. And this water use climate influence, climate also in current year temperature and previous year precipitation influence the water use efficiency. Water use efficiency in tuna ciliata was affected by previous year June temperature, and Lagerstomia speciosa was negatively connected with current year June temperature. And these are the interannual variability was mainly driven by the temperature. Long-term increasing trend in water use efficiency was explicitly contributed by atmospheric carbon dioxide, that means Ca. Mostly contributed Ca, the atmospheric carbon dioxide. And also oxygen isotope, we also measured the oxygen isotope, delta 18O and delta 18O annually, how delta 18O varied like in 1985 to 2015. And it showed a strong correlation with most of the month of the PDSI, also BPT, precipitation and temperature maximum. And this one mainly for oxygen isotope, plant functional types matter. That means Cicracia tabularis is a partial shade tolerance, tuna is a light demanding, Lagerstomia is a shade tolerant species. And tuna has a high delta 18O than the others too, Cicracia and Lagerstomia species. And now, as you know, the Bangladesh is a very extreme, have a lot of extreme climatic events like a drought and flood. Drought, there is a several drought in faced Bangladesh. That means 1972, 78, 81, 82, 84, and recent also increasing the intensity of drought. Also, we have a flood events in Bangladesh. We selected two drought year, 1999 and 2006. And in comparison of the normal years, the drought years had longer dry season, short growing season, low precipitation, higher mean, minimum, and maximum temperature. And these drought events, two drought events, how matter with the basal density. That means percentage change of basal density. In drought year, number of basal, total basal area, and mean basal area, basal density, basal area 50% decrease. However, basal density slightly increased. And SPI is a recognized drought index. We correlate the SPI with ring width, number of basal and mean basal area. And we found a very good correlation with SPEI. That means SPEI, a drought also influence our basal also ring width, negatively impacted. And this also we find in the spatial correlation function. And another drought years, 1984 and 1999, we have seen the ring width decrease in both species, Cicracia tabularis and also Tuna ciliata. Drought year 1984 and 1999, they showed the decline growth and 50% comparison to normal year, it almost 50% decrease in growth. And this impact how the resilience factor, I think you know some resilience index like a resistance, recovery, and resilience, and relative resilience. These are the four parameter factor. By this four factor, we can see the how drought impact on tree growth or basal growth, tree growth. And we have seen no significant difference in drought resistance. These are the resistance, first one. And Cicracia tabularis showed higher recovery. And drought resilience differ between species and drought sensitivity. Cicracia tabularis is more drought resilient in severe drought. We in Bangladesh also we have some flood events, and we can do the flood event analysis by using the dendrochronology. There is a major three floods, 1974, 1988, and 1999, 1998 and 88. And these four floods, also there is, in this figure, you can see the Monu River, Surma River, and Kuai River, all of them water level also decrease, some in flood level, that means it increased. And then how these three flood events influence in our growth. Like in 1975, look at the generally the growth ring, growth ring width is more, but in flood event, it also reduce the growth. And most of the flood year, they showed the negative growth. And look at the ring width and vessel chronologies, like a ring width chronology, number of vessel chronology, also vessel density. And one important thing, that means delta 18O also showed this flood signal. Delta 18O also showed this flood signal. And flood year, the 3 flood year, they showed the delta 18O, the 2 flood year, they showed the negative influence in delta 18O. And these are all the things about the Bangladesh dendrochronology. But we have lot of species in Bangladesh to study the dendrochronology. Especially, we have a lot of species who showed the distinct growth ring boundaries. That's why we have a lot of opportunity to do dendrochronology more in Bangladesh. Also, in different aspect, we can focus different aspect in dendrochronology in Bangladesh. And if the tree from the ring with distinct ring width, also we have to check that if it is possible to cross dating, then we can make a chronology. But also we tested some species, that means we have the opportunity to cross date among some trees can be cross dated, some species can be cross dated. Then we can also use this species for future study. And for also we can estimate the age of the different species and different tree level by using the dendrochronology method tools. And that means huge potential in Bangladesh. We collected a lot of cores. Also, we can examine, we already did this, some examination of tearing with measurement and tearing with detection. Then it's a huge potential of tearing analysis in Bangladesh. Yeah. Thank you so much. And especially I want to thank my university. Also, I want to thank special thanks to my supervisor, PhD supervisor, and also, all the members, all the participants who helped me a lot to do the tearing research in Bangladesh. Thank you so much.
Okay. Thank you so much, dear Dr. Mahmouda. And if our assistant have any question, please, you can do this. Okay, we have question here in the chat.
Yeah.
Basic say, thanks for this presentation, and have the following question. Is there any application of tree ring research in climatic reconstruction?
Yeah, this is nice questions. I think I like the idea of reconstruction. We had a plan to reconstruct the tree in Bangladesh, but one important problem in Bangladesh, because we have not enough tree, is a big tree like a more than 100 years, 300 to 400 years, 500 years trees. We don't have that much, but we try to get at least some trees to do the reconstruction.
Okay, thank you so much, dear Dr. Mahmouda. Any participant have other question, maybe? Okay. Aquí tenemos, we have other question. Oh, congratulations, say. Their target say, Salaman, nice presentation, best of luck for future research. For you, dear Dr. Mahmouda.
Yeah, thank you. Thank you.
And Okay, I need to see. Okay, our participant is the English language and foreign language. Thanks, this is the part of the finish of our presentation. And there, Dr. Mahmouda, thank you so much for this opportunity.
Thank you so much. Yeah, thank you so much for
Thank you
yeah.
so much
I
for
can share my research to you also. Yeah.
for meeting again.
Yeah.
And
Yeah.
always I remember you and your family.
Yeah.
Very amazing.
Thank you.
And I desire and I wish that you following obtain more a good results about your research in other opportunity we call meeting again.
Yeah.
And
Yeah. Okay. Thank you. So
a strong act for you. Thank you so much and
yeah,
see
thank you so much. Yeah, is Dhanyabad, gracias.
Gracias.
Yeah.
Sí, muchas gracias. Dankeschön.
Yeah, danke schön. Yeah, danke schön.
Thank you so much and best regards for
Yeah,
all.
best regards.
Thank
Best
you so
regards to
much.
you. Yeah.
See you.
Yeah, see you. Okay.